Today, I will illustrate how the scientists Nernberg and Mattei discovered that amino acids are coded for by unique RNA sequences, and additionally, I will walk through how the scientists Nernberg and Leader went on to discover what different combinations of nucleotides coded for which amino acids. And since it came first chronologically, I'll start with Nernberg and Mattei's work. What they did is they started with a homopolymer strand, which is a strand consisting of only one repeating nucleotide. And they combined that in a tube with the components of a cell called cell-free extract because it's cell content outside of the cell itself. And in their first experiment using this technique, they used a poly-U strand, which is a uracil homopolymer. And they did 20 trials with the solution of poly-U and cell components, and in each trial, there was a different amino acid that was radio-labeled. So once they allowed translation to happen, they would find 19 normal-looking solutions and one solution that had a radioactive protein. And this implied that whichever amino acid was in this tube was coded for by the codon UUU. And this amino acid happened to be phenylalanine. And they also did additional research about poly-C, which codes for proline, poly-A, which codes for lysine, and poly-G, which they didn't actually find an amino acid for. They didn't find anything that associated with it. But this progress allowed Nernberg to go on and further study which amino acids were coded for by which nucleotide sequences with another scientist named Leader. And their procedure was to combine small pieces of RNA consisting of different combinations of nucleotides, like for example, I could use CGU. And they combined this with radio-labeled tRNA, the squares, attached to amino acids, which are the circles. I have arginine and serine here for examples, but they would have all 20. And then they combined that with ribosomes so that they could all bind together. And similarly to the experiments with Mattei, they did 20 trials for each suspected codon and radio-labeled a different tRNA amino acid each trial. If, when the mixture was passed over filter paper, a radioactive substance was left over, they knew that whichever amino acid was combined with the solution was coded for by whichever codon they used in that set of trials. Like this example, the CGU was the mRNA sequence that was able to bind with GCA in the tRNA, which is associated with arginine. And since radioactive substance was left over, they were able to see that CGU codes for arginine. And obviously this wasn't the first exact example they used, but this was one of them because this led them to make the coding dictionary possible, which is an index of amino acids and what combinations of nucleotides could possibly code for them.